Welcome! In this lesson, we'll build an intuitive, physics-based picture of how cells use adhesion domains to assemble biochemical reaction platforms. We'll see the tug-of-war between short-range attraction and long-range repulsion, how immunological synapses control enzyme access, and how Marks and kinases orchestrate growth of philopodia. First, the physical picture of adhesion. Two neighboring cell membranes face each other. Short-range attraction is created when pairs of cell adhesion molecules form links, but long, flexible glycoproteins in the glycocalyx generate a steric repulsion that keeps membranes about 50 nanometers apart. The observed adhesion domain is a local compromise, a cluster of strong links surrounded by weaker contact zones. Let's draw two membranes and the glycocalyx. Now we nucleate a tight adhesion domain by clustering adhesion pairs. Finally, the cluster is stabilized from the inside by coupling integrins to actin patches through talon-like adapters. Now, a beautiful example, the immunological synapse between a T cell and an antigen-presenting cell. Before adhesion, a large phosphatase called CD45 reaches the T cell receptors and removes phosphate groups, keeping adapters silent. After a tight contact forms, the bulky CD45 is sterically excluded from the narrow gap. Kinases can then phosphorylate T cell receptors and recruit adapters, activating downstream enzymes like PLC gamma. Let's draw the contact and show the non-adhesive state first. Now we form the adhesion domain and exclude CD45 to allow activation. Next, a molecular gatekeeper story at the inner membrane, the polybasic protein MA. Our CKS shields negative Pi45P2 lipids, preventing random enzyme binding. To trigger local reactions, our CKS must be displaced. Strongly binding Pi3K can start the displacement, producing Pi345P3. PLC can then generate DAG lipids that recruit PKC, which phosphorylates CKS and speeds its unbinding. This cascade can launch actin bundle growth and myosin 10-driven transport of integrins into a finger-like filopodium. Let's sketch an inner membrane patch with Pi45P2 and the CKS shield. Now a strong binder, Pi3K, arrives and locally displaces CKS to make Pi345P3. Next, PLC produces DAG. PKC binds and phosphorylates CKS, further unbinding it. Finally, actin bundles push a protrusion, while myosin 10 motors carry integrins to the tip. A final systems level note: T cells often need repeated, discrete synapse engagements before committing to division and antibody production. This is called a quantal law of immune response. Each synapse is like one counted pulse toward a threshold. We visualize this as counted pulses ticking a tally toward activation. Summary: adhesion domains emerge from a balance of short-range binding and long-range steric repulsion, and they are strengthened by integrin coupling to actin. Immunological synapses control biochemistry by physically excluding bulky phosphatases, tilting the balance toward phosphorylation and adapter recruitment. Inside the cell, CKS shields negative lipids until strong binders and PKC phosphorylation displace it, enabling localized lipid signaling, actin bundle growth, and targeted delivery by myosin 10. This is how physics of membranes and forces guides where and when reactions can happen in cells.